So the reason why we watch these clips, we don't really have a wet lab that kind of like gives you a sense of how this work is done. And even the simulations on PhysioX, they don't really give you a sense either. I don't even assign them. These clips that I found they give you some idea of what these, the work that Hodgkin-Huxley did. And so I want you to appreciate the squid giant axon, and you just watch them clean it here. Let me see if I can get it going.
All the small fibers surrounding it have first to be removed. In order to expose the giant axon itself, all the small fibers surrounding it have first to be removed. This is called cleaning the axon, one end of which is often left with the small fibers attached for handling purposes while the axon is prepared for the experiment.
I can't hear it, Professor. Is there any way to make it any louder?
It usually takes about half an hour to clean an axon and to leave it bare of all the small fibers, often over a distance of about 5 centimeters.
Let's run it back, and I'll turn up the volume here.
This is called cleaning the axon, one end of which is often left with the small fibers attached for handling purposes while the axon is prepared for the experiment.
Yes, no problem.
It usually takes about half an hour to clean an axon and to leave it bare of all the small fibers, often over a distance of about 5 centimeters. When finished, the giant axon alone remains, a single animal cell several centimeters in length and, in this case, about 650 microns in diameter. Each small division of the scale is 100 microns. A cell this size weighs about 20 milligrams, so that it is not surprising that the giant axon was first used as a source of cytoplasm. In 1937, Baer, Schmidt and Young, working at Woods Hole, studied the protein constituents from samples which they obtained simply by squeezing the cytoplasm out of the cut end. Later workers made precise measurements of the electrolytes and found that the concentration of potassium was much higher and that of sodium much lower than in the surrounding body fluids. It was for studies of the viscosity of cytoplasm that a cannula was first inserted into the axon, and it has since proved such a valuable tool that it is now used routinely. A thread is attached at each end of the axon and pinned down to hold it taut. In addition, 2 threads, which will later secure the cannula, are slackly tied round it. The cannula itself is a carefully drawn piece of glass tubing, the end of which has been ground down to form a smooth, oblique tip, slightly smaller than the diameter of the axon. The next step is to cut, without severing the axon, a notch in its wall large enough to accommodate the cannula. A micromanipulator provides the fine adjustment needed to pass the cannula through the notch and some distance along the axon. Once in place, the loop of thread is brought up and the cannula secured. Finally, the threads are trimmed and the uncleaned end of the axon removed.
All right, so that's the setup. But they use the biggest cell so they can get their little electrodes in there. Okay, 650 microns in diameter. Here's the other one.
An electrode was first placed inside the axon by Hodgkin and Huxley in 1939 at Plymouth, and by Curtis and Cole in 1940 at Woods Hole. Hodgkin and Huxley first made a plastic cell and mounted it on a platform which could be raised and lowered like a lift. As Professor Baker shows, the axon held by the cannula was then hung in the cell which was filled with seawater and connected to an external electrode. The internal electrode, which was not attached to the lift, was then placed vertically above the axon, with its tip in the cannula. It was centered with respect to the cannula and axon by placing a small mirror beside the axon, arranged so that a second image at right angles to the first was seen through the microscope. By adjusting the position of the cell, both horizontally and vertically, the cannula and axon were now raised up over the tip of the electrode, which always remained in the same line of sight. Hodgkin and Huxley found that as the electrode entered the axon, a negative potential with respect to the external seawater of about 65 millivolts was obtained. This was the resting potential of the axon, and although its existence had long been suspected, this was the first time it had been directly measured. Moreover, when the axon was stimulated, the action potential did not simply fall to zero during the impulse, but became positive with respect to the outside, shown by the overshoot of the action potential. This important discovery suggested that the nerve membrane, which at rest is mainly permeable to potassium, becomes primarily permeable to another ion during excitation. This other ion is sodium, since if its concentration in the external solution was lowered, the action potential immediately became smaller, by an amount depending on the sodium concentration. If, as these experiments suggest, the action potential was dependent on the passage of ions across the membrane, it was obviously important to measure the currents carried by these ions. To do this, it is necessary to hold the internal potential at a chosen value.
Okay, I'll stop it there. Voltage clamping, you don't have to understand that. Up until I showed it, you should understand it. And I think it's not too hard a concept to see, well, if you remove sodium from the external solution, the magnitude kind of shrinks, right? Because there's not as much to influx, so the membrane potential doesn't change as much. Okay, so that does it for the clips. And let's get back to the PowerPoint. So these 4 questions. What was the diameter of that giant axon? What was the number?
665.
Yeah, 650, but what was the units there? The microns. Number 2, the sodium-potassium concentration in the cytoplasm compared to the surrounding body. I mean, just what we've been talking about, right? What about it for potassium?
Higher.
It's higher and lower for sodium compared to the outside. And what was the resting potential of that split giant axon when they measured it? Negative 65 millivolts. Well, I just answered this one. When they removed sodium, the action potential became smaller. All right, so let's move on. Now let's make sure we covered all these properties of the action potential. We went through the different phases of the big spike. And I have touched on some of these, not others. Properties of the AP, which stands for action potential. Number 1, threshold must be reached. I shouldn't have erased it, but what I had on the board before. In millivolts. If negative 55 or negative 60, something like that, is the threshold. You must reach it. Okay, it's like an off-on switch. You know, off, off, and then if you reach threshold, it's on. And you get the full spike there. So off, off, on. Threshold must be reached for it to be turned on. And that kind of illustrates number 2. It's all or none. Either you get all of it, the full action potential big spike, or none of it. Okay. And when you get the full thing, that action potential will fire and it'll stimulate the next cell. And number 3, refractory period. The refractory period, refractory in general means it's a period of insensitivity. In this case, it means you cannot get another AP. A period of insensitivity. Well, they're kind of like, it's kind of a gray area here. There's what's called absolute refractory period and relative refractory period, and you should know the difference. Absolute refractory period. Let me go to the next slide here. Here's our action potential, and they have kind of color-coded absolute versus refractory period. Absolute in green, right there. There's the term. Kind of this part of the action potential is basically absolute refractory period. You absolutely cannot get another action potential, simply because one is occurring. So I'll just speak plain here. You just can't get another AP. So what's happening is the cell is either in a state of depolarization, right? It's either depolarization or it's when the sodium channels are inactivated. During sodium channel inactivation, you can't get another AP. Sodium channel inactivation. Now the other one, a relative refractory period, relative refractory period. You can get another AP if the stimulus is big enough. So let me continue up here and get into the bottom of the board. Continued. You can possibly get another AP if the stimulus is strong enough. So it's kind of the dark blue portion of the action potential trace. Right there, kind of the after hyperpolarization thing. So this is during the hyperpolarization. The cell is being hyperpolarized, this relative refractory period. During hyperpolarization. During that time, the sodium channels are no longer inactivated. They're in their rest state. However, you still have some potassium efflux going on. So you do have some potassium efflux However, the sodium channels are in their rest state. Quote unquote. That means something in particular. They're not inactivated. The activation gate is closed. That's what that means. I defined that before the break. And so, what let me kind of see if I can draw this here. So it's like got a threshold potential. And let's say we reach depolarization, repolarization, hyperpolarization. And let's say right now is when you want to re-stimulate. You can, but you need a bigger stimulus. Before, I only needed a depolarization to get me that far. Now I need a depolarization to get this far, right? It's like, let's say if you're way hyperpolarized, you need a bigger stimulus. Is the point, right? Because you're further away from threshold. So that's the relative one. Excuse me. Drink of water. Okay, the other thing I said about this refractory period, it ensures one-way propagation down the axon. Axons travel in one direction, not two. It's one way toward the axon terminal. Refractory period ensures one-way propagation of the AP. So to kind of illustrate this point, it helps to kind of like think of the axon in segments. Let me erase this top part here. So let's say you have a big axon terminal way down here on this side, but way at the beginning on this side, we have our first segment of the axon where you get an action potential. So let me draw kind of the axon in segments. I'll draw the first few segments there. So each is a segment of this long process called the axon, and this is the axon terminal. The goal is to propagate the action potential from each segment all the way to the end here. So relative refractory period ensures one-way propagation so it doesn't go in reverse towards the soma. Because let's say, for example, you stimulate here, and you get your influx of sodium. So you're depolarized, right? You get your action potential, your spike. And then, so it kind of, you look down the tube and you see it's all negative, okay? It's all negative. And so you go to the next place where it's negative because positive is attracted to negative. So then these become positive. You kind of like travel from here to the next segment and you depolarize and get an action potential in the next segment. In the second segment, now you kind of have a decision now. You could go back to the first one or to the third one. So it looks backwards and it sees that the previous segment is in some state of refractory period. Okay, it's still positive there. It prefers to go where it's negative, so it's just going to go one way. So, so this is this first segment in refractory. So it goes, okay, I'm going to go to this one then, because it's still negative here. So you depolarize the next segment, and you get an action potential. And now you have a choice. You can go backwards or forwards. Well, the previous segment is always going to be in some state of refractory period. So you just keep going one way. Okay, so that's why refractory period ensures one-way propagation And that's also why you get this non-decremental conduction. What that means is, as you get propagation of the action potential and you depolarize each segment, if you could measure it at each segment all the way to the last segment, the action potential does not lose magnitude, okay? Because at each segment, there's voltage-gated channels that you keep turning on to get your full action potential at each segment, okay? Because let's remember, when we study the action potential, it's measured at a segment. You're not measuring the whole tube, okay? So what we're saying is, as it does travel, you don't lose magnitude. That's the next one. Number 4, non-decremental conduction. The AP does not lose magnitude as it propagates down the axon. Because there's voltage-gated channels along the whole thing. Doesn't have a chance to die out. Voltage-gated channels are along the entire length of the axon. So compare action potential, non-decremental conduction with graded potentials. Go back and look at your notes. I specifically made a point to say that graded potentials are decremental. Action potentials are not. So that's one of the contrasts we like to bring to students. So the next point, at what speed does the action potential propagate? What's the speed? What's the velocity? What's the miles per hour? Well, you have continuous propagation and you have saltatory propagation. So again, let's kind of think, how can you study that? It helps to think of the axon in segments. Just to go from segment to segment, you're traveling. Let's say you travel from there to there, some distance x, to go from just from one segment to the next one, okay? And so the question, how I want to teach this is, what determines the length of x? Because that will determine the speed, the way you can go to x. And I always ask this question, and intuitively you think the opposite. So let me, okay, let's say x is shorter. Let's say x is longer. That distance from segment to segment. if x is shorter, is the velocity faster or slower? Students like to say faster, and that's wrong. That's where it's like, I'm not trying to trick you, it's just you always answer wrong every year I ask this question. It's okay. Year after year, you guys mix it up. Shorter is slower. It's like, if you gotta go like, ding, 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 slower. But if x is longer, you go boom, 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 boom. It's much faster. Okay, so again, what determines the length of x? That determines velocity. Axon diameter. Okay, let's think about that. So if you look at an axon in cross section, say a smaller one versus a bigger one, usually a source of resistance to velocity is contact with the cell membrane, usually. So usually if you have a bigger space to travel down, that speeds up velocity. So the general rule is a larger diameter means you're gonna have faster conduction velocity. That's what we call it. This one would be faster conduction velocity. To give you an idea of how fast, I mean, there's a range of 2 to 200 ballpark miles per hour, okay, that these things can travel. Axon diameter is one determinant. The other is, well, I say leak. Maybe you have a break in the membrane or something. Well, if you have a leaky membrane, and it's like every time you try to depolarize, positive leaks out, it's more difficult to reach threshold, so that will slow down the velocity. So the rule is, if you increase leak, more difficult to reach threshold, slower velocity. Okay, the other thing is myelination. Is the axon myelinated or not? If the action is, well, I drew this unmyelinated. Okay, let's say I drew the same thing, but myelinated. Let's try to draw the same one here. For this one, I'm gonna put these like big old myelin sheaths around it. So those are our Schwann cells with our myelin. And the gaps between them are those nodes of Ranvier. Node of Ranvier. So where you have the myelin, it's cell membrane. It's kind of fatty cell membrane. You can't have any influx there. There's no, you can only have the voltage gated channels at the nodes. So you only have them here at the nodes. So let's say you depolarize the first segment. The next place you could depolarize is at the next node. So you have to jump all the way to here. okay, and then depolarize there. And then jump, and then to the end, right? So X is much longer and the conduction is faster. Ooh, 1 second here. So you give me a chance to process this. Where's my pen? On your half sheet. We got to number 4, right? So we're on number 5, is that correct?
Yeah.
Number 5, yes, thank you. Explain. Oh, I got some explaining to do. Explain. It's our favorite word in the profession. Your least favorite word when you have to explain something to me. Right? Explain why refractory period ensures one-way propagation. That's number 5. Number 6, you tell me, what determines the length of X? There's different things I just got done talking about. So work on that for a couple minutes. We'll check back in. Write small, use the front, use the back if you have to, of your little half sheet there. And All right, let's continue on. If you're still working on that, I'll give you time after the lecture to finish. But I wanted to show you one of the things that determine the length of X. I kind of illustrated here. The myelination really picks up the speed. We call this form of propagation saltatory propagation or conduction. This is saltatory, which means jump. The signal is skipping from node to node. This one is referred to as, without myelination, continuous propagation. Or it's illustrated here in animation. This is myelinated, and the impulse is traveling much faster. Here they don't even illustrate it getting all the way to the end. I think they show it like a race. It's like this one always finishes, this one get barely started. Okay. Anyways, myelination makes a big difference in the conduction velocity, amongst the other things that we listed for you there. All right, so those are all the properties of the action potential I wanted to talk about. Yeah?
Did you say the myelination makes it faster? Because of the jump?
Mm-hmm. Conduction velocity way faster with the myelination. So this slide reminds students that when we study the action potential, we're only measuring a particular segment. And as it travels by, we catch it. Okay? So before it gets there, all you record is rest. But as it gets there, you first depolarize, followed by a repolarization. Okay? So we're only looking at one segment. But let's remember that it's propagating down the entire length of the axon because there are voltage-gated channels throughout its length. Okay? Look at the top graph here. You have a cell. Look at the axon. If you didn't have voltage-gated channels along the whole length, it was just a bare plasma membrane, you stimulate, get a big signal, but it dies out immediately. It can't propagate without the voltage-gated channels. But if you do have, say in this example here, at each segment, voltage-gated channels, it'll propagate, it'll jump along there. It'll really jump with saltatory conduction when you have the nodes of Ranvier, the gaps, that fill basically the myelin there. So this is a much longer length of X for the fastest conduction. So that's how the signal propagates. The goal is to get to the axon terminal. This electrical impulse must reach the axon terminal so that a chemical signal can be released into the synapse to stimulate the next cell. And how you stimulate the next cell is another topic here. The book calls it information processing. Okay. At the simplest level, neurons, dendrites receive neurotransmitter messages simultaneously. Some are excitatory, some are inhibitory. So the effect on the axon hillock determines if an action potential is produced. So this is kind of talking about what I showed you this slide before. When the action potential propagates down and finally arrives, what's going to happen here? Well, we studied this already. ACH is released and it stimulates the next cell. So that's why these terms are used. EPSP, IPSP. Here's an axon. Here's an axon terminal. Here's a cell. Another cell is trying to stimulate. Okay, I'm just basically trying to draw that little picture there. But okay, so if this is my synapse, is this the presynaptic or the postsynaptic cell, the blue one? It's pre. So what's the black cell? Post. And how you stimulate the postsynaptic cell is what we're talking about. If the term EPSP is used, read what it says on the slide, excitatory postsynaptic potential. Okay? It could be excitatory or it could be inhibitory. IPSP. So postsynaptic potential means how are you affecting this cell, the postsynaptic cell, not the presynaptic cell. Okay, well, this is not new information, just the terms are new, I guess. Here's a picture of an EPSP. It's a depolarization that brings you closer to threshold. Dipole. You know, anything that brings you closer to threshold is considered excitatory. It's excitatory, that's the E. It's a depolarization, it's an influx of sodium, probably. Or if you have an efflux of potassium, look at that. That little dip there. That is an IPSP. So it's a hyperpolarization. Brings you further away from threshold. In the postsynaptic cell, that is. So, in these two scenarios, EPSP, IPSP, which are you more likely to get an action potential? EPSP or IPSP? More likely to get an action potential. EPSP. You get closer to the threshold, you're more likely to get an action potential. That's considered excitatory in how the postsynaptic cell is processing the information. That's BIOL. So it's basically to reach or not reach threshold, right? How, you know, if you want to trigger an action potential, EPSPs should do it. One EPSP, like I say on the slide, may not be enough. EPSPs, IPSPs, they basically add up via summation. And this is what we call information processing. You're reaching threshold or not. And there's two kinds we define, temporal and spatial summation. Let me write that down first. EPSPs and IPSPs, quote unquote, add up. You either reach threshold or you don't. And the way they add up is referred to as temporal spatial summation. Temporal summation is like one input fires twice with rapid succession. Okay. One input, one synapse, I should say, fires twice in rapid succession. Bam, bam. That's why the word temporal is used versus time. It's just boom, boom. But it's one input, so it's one cell firing twice onto the postsynaptic cell, trying to stimulate it. Okay. Whereas spatial summation, it may be more than one input. It could be 2 or 3 inputs, maybe many more. Spatial summation. I'll just say more than one input. Each fire simultaneously in different locations. More than one input. Fires simultaneously at different locations. And those firings can either be EPSPs or IPSPs, and they're going to add up, and you'll either reach threshold or not. So we like to show you data and have you analyze that. And look at that. So are we on number 6? Number 7 on your half sheet?
7.
We're on 7. Number 7. Look at this data. I want to show you similar figures on the exam. And let me ask you some questions here. Let's see. Was threshold reached? Did you reach threshold? Did the cell, not you, did the cell reach threshold? That's a question. Call that 7A. That's a yes-no question. B, where the signals excitatory or inhibitory? Were there EPSPs and or IPSPs? Lastly, was there any kind of summation? Was there any kind of summation? That's the question. If yes, what kind? All right, answer 7A, B, C, according to these data on your half sheet. Go, give you a minute. Talk about it with your neighbor if you're stuck, or just quiz yourself if you think you got it. All right, I don't consider these difficult questions if you understand what's going on. Let's make sure we're all getting it here. A, did you reach threshold? Yes or no? No, you did not. There's no action potential here. You didn't reach threshold. It looks like, you almost did, but you didn't. No action potential, you did not reach threshold. Oh. Were there IPSPs or EPSPs? What do you think the E stands for? EPSP. So only 2 EPSPs. And do you see the input there, that green cell? Right there, E1 refers to that cell firing on the yellow cell. So this cell I'm touching, it's not on your half sheet, but is this the pre or post synaptic cell? Post. This is post. That E1 input is the pre. Okay. All right. Was there any kind of summation?
No.
Nope. It says that. That was the easy one. All right, number 8. Same questions. Just change that to 8. New answer. Give you a minute for these data. Yeah. That's correct. I think these 2 are, it wasn't rapid enough.
Oh.
Too far apart. Moving on to the next one. All right. Did the cell reach threshold? The answer is yes, because you got an action potential, right? You got a big spike. Were there EPSPs or IPSPs? Which one? There's definitely 2 EPSPs. Bam, bam. I still see the same 1 input, but this time it fired twice in rapid succession, whereas the prior slide wasn't quite fast enough. What happened prior was you just spaced them. You allowed the membrane potential to fully repolarize, and you fired again, didn't do nothing. But when you kind of stacked them up, they summated. You had enough depolarization to reach threshold. Okay, that's what that looks like. So the answer for letter C was temporal summation, right? 1 input firing quickly. Okay. Yeah, look at this one. Okay, you're on number 9. All right, I think that was enough time. Did the cell reach threshold? Yes or no?
Yes.
It did. I see an action potential. Were there EPSPs or IPSPs? Yeah, and they were both excitatory. And this is spatial summation. Do you see more than 1 input? Yes or no?
Yeah.
I see an E1 and an E2, assume they're firing at the same time, and they add up. Okay?
And those both are considered pre still, right?
Yeah, E1 and E2 are considered pre. That is correct. So yellow cell, post. Okay, number 10. Never mind. I think you get. I hope you get it.
Yeah.
Let's just speed it up. But now we have. What does I stand for?
IPSP.
IPSP. So that makes sense why it kind of like hyperpolarizes a little. What happens when you add it with the EPSP? It's like they kind of cancel each other out. Do you reach threshold? No. Is there summation? Yes, there is. They just kind of cancel each other out because one's an IPSP. See how that works? The bottom line is you inhibited the cell. Maybe you wanted to do that. Okay. So that that red one, that input is the causing the IPSP. So here they are side by side. And this is the subject of information processing, temporal spatial summation. And so basically to redefine temporal, just one fires rapidly, okay, or spatially, you got two firing simultaneously, okay, as may have the same effect as one firing rapidly. That concludes the material for your exam, which will become available on Proctorial Friday. When you come to class on Friday, we will start the next unit for the vertebral column. Try to get those that next unit available for you. For today's lab, there's not much time left, but it's just that worksheet. And I think most of you have it done. Since there is some time left and you may turn it in, I'm just going to say nervous tissue lab due today. Only what I covered. So for the nervous tissue lab, turn that in today. And also, a lot of people are asking me about the multiple choice exam. I haven't finished the written part. I usually like to give it back together, but since so many of you are anxious to see how you did, I'll give you a chance to review the multiple choice part today. And Friday, I'll let you look at the written part. I'm almost done with it. Okay. And so if you want to see the multiple choice part today, stick around, give me some time. I'll get them out and get an exam up here so you can look and see how you did on that. And just be sure you turn this in today. Make a pile by the torso like we always do. And don't forget to turn in your half sheet as well.
Professor? Sorry.
Yeah.
Who was this? We were just